Thank you for that kind introduction. Can you all hear me? Can you hear me?
Yes,
okay. Can you see the slides as well?
Yes, sir.
Okay. Thank you. So, thank you, in fact, for inviting me. Thank you, Rishikesh, for inviting me to this global Ayurveda.
Can you shift into that presentation mode, sir?
In
PPT mode.
what do you want me to do at my end?
The screen with the
slideshow, sir.
Slideshow.
Can't you see the slides?
It is in the slideshow mode.
Just 1 second.
No.
Right. Okay.
Yeah,
it is in the slides. Okay. Can you see that? Yeah. Okay. Thank you. So thank you, Rishikesh. Thank you, in fact, the organizers for inviting me to this Ayurveda conference. And so what I'm actually sort of going to do is give you an overview of natural products and Ayurveda in the prevention and treatment of atherosclerosis. So for the last 20 odd years, I've been working on atherosclerosis. Atherosclerosis is an inflammatory disorder of medium and large arteries associated with deposition of fats and cellular debris. And what this can do is cause narrowing of the arteries, impeding the blood flow. And in its mild format can give you sort of symptoms such as angina. But the clinical complications of atherosclerosis arise when so-called atherosclerotic plaque rupture. And depending on the site of rupture can give rise to cardiovascular diseases such as myocardial infarction, heart attack, cerebrovascular accident, stroke, and peripheral vascular disease. It's a major killer worldwide. In the UK, we have reduced it in the last 7 to 8 years from 33% to 25%. Cancer, in fact, overtook atherosclerotic cardiovascular disease about 3 years ago. It's obviously a major economic burden. Total cost to the UK economy is around £9 billion a year. Globally, it's even worse. Accounts for around 31% of all deaths, and on the increase in several countries because of acquisition of a westernized lifestyle. And in fact, whenever I actually visit Bangalore, I realize, in fact, the influence of this westernized lifestyle in the malls, right? The queue around KFC and McDonald's, in fact, is much bigger than the queues, in fact, for those who want to eat idlis and dosa like myself. So clearly, in fact, this acquisition of westernized lifestyle has created a major problem in some countries. And of course, it doesn't help that statistics are extremely depressing if you happen to be a South Asian like ourselves. Now, in India, it's only recently that we are actually gaining more insight into the alarming statistics. And still, in fact, this probably represents it's an underrepresentation. So in 2016, around 28% of deaths were from cardiovascular disease, and this increased to around 33% in 2020. What, of course, is more alarming is that 59% increase from 1990 to 2010. And also more disturbing that this occurs about 10 to 15 years earlier than in the West, with an accelerated buildup and a high rate of mortality. So my first visit to Bangalore was in 2013, where I actually attended the Thrombosis Institute when she was there. And of course, the statistics there were really quite alarming, in the sense you got individuals in their 30s and 40s dying of heart attacks. Obviously, I'm a big fan of Bollywood, big passion, in fact, for Bollywood movies. And in fact, in the short time that I lived in India, 3 to 4 years, I watched around 500 movies. In fact, one thing which I wanted to do was be a singing Bollywood star. Of course, that didn't materialize, and I finished up being in academia in the UK. But here are some gems in Bollywood that we've lost due to cardiovascular disease. So Mukesh, Mohammed Rafi, Kishore Kumar, obviously all died of heart attacks. Sanjeev Kumar, Vinod Mehra died of heart attacks at the age of 47. And more recently, David Nischal and Om Puri. So clearly, it has actually had a major sort of impact on sort of worldwide, and obviously in India because of this accelerated buildup which occurs at an early stage. Now, it's a multifactorial disease having non-modifiable and modifiable risk factors. And in terms of the non-modifiable risk factors that we are all aware about, obviously it's a disease of the aged population. Male gender has got a high predisposition. But of course, there's all this genetics in terms of personal and family history. And this is where the South Asians are more predisposed to the sort of mutations, which of course increase risk factors for this disease. And this, of course, is coupled with modifiable risk factors, such as smoking, a high level of plasma LDL cholesterol, high blood pressure, diabetes. Of course, India is regarded as a capital of diabetes worldwide, obesity, lack of exercise, and pollution. And it's obviously this combination of these non-modifiable risk factors plus multiple modifiable risk factors, which of course has created the sort of statistics, disturbing statistics that we're seeing in the South Asian population. And of course, the risk factors act in a synergistic manner. So if you're just a smoker, you've got a 1.6% or 1.6-fold increase in developing cardiovascular disease. But if you're a smoker, have hypertension, and also high serum cholesterol level, then this actually increases to a massive 16%. Now, of course, lifestyle changes is a key contributor to prevention of atherosclerotic cardiovascular disease. And one of the things which has, there's a sort of educational drive right from the primary school in the UK of educating the population, younger children in particular, to have healthy lifestyle, a healthy diet, healthy living, healthy exercise. But of course, once the disease has kicked in, one needs to take pharmaceutical drugs. And this could be either anticoagulants, aspirin, for example, to prevent plaque from rupturing, beta blockers to actually normalize the heart rate and also reduce blood pressure. And in terms of the blockbuster drug, it's statins. And statins are inhibitors of a rate-limiting enzyme in the biosynthesis of cholesterol. And of course, cholesterol is a culprit, particularly LDL cholesterol in atherosclerosis, because it causes a state of chronic inflammation. And statins, in addition to inhibiting de novo cholesterol synthesis, and bear in mind that 80% of the cholesterol is made by your body, statins also have other pleiotropic effects. For example, it acts as an anti-inflammatory manner. And so that was one of the comments that I actually had, that individuals who are on statins have been protected against the deleterious effects of COVID-19. Statins, of course, have other sort of pleiotropic effects. For example, they stabilize endothelial cell dysfunction. So clearly, statins have been extremely effective. But if you were to ask one question, how much of the recent reduction in cardiovascular mortality that we have seen in UK and other countries in the West, how much of that you can attribute to statins? The answer to that is 30%. So in other words, 70% of individuals on statins still die of heart attacks. Not only that, statins have got other adverse side effects. They affect the muscle, the liver, and in a younger population, they predispose them to diabetes. So whilst there's a wealth of evidence from clinical trials involving millions of participants on the effectiveness of statins, clearly there are major issues. So we need to actually look at alternatives. to statins, particularly for those 70% of individuals on statins who will still finish up with cardiovascular events. So beyond statins, there have been few successes. One of them is shown here, ezetimibe. Ezetimibe is an inhibitor of a transporter of cholesterol in the intestine. And so this targets the dietary cholesterol, which accounts for about 20% of the cholesterol in the body. And clinical trials of statins plus ezetimibe have shown successes. One of the hot target at the moment in the cardiovascular field is this protease PCSK9. Now, what happens is that in the cells in peripheral tissues, you got LDL receptors. These LDL receptors will pick up the LDL particles, and LDL, of course, is regarded as a bad cholesterol. They'll pick up the LDL particles and they'll undergo endocytosis. This will actually finish up in the lysosomal compartment, where the LDL particles will be digested and the receptor will recycle back to the cell surface to pick up more LDL cholesterol. What PCSK9 does, it causes modification of the LDL receptor so that instead of recycling, the receptor gets digested as well, along with the LDL particle. So there's little recycling, so the concentration of LDL receptor decreases on the cell surface, and so you got less clearance of LDL particles. Now, so far, since 5 or 6 years, in fact, when of course PCSK9 really came into the scene, we have not been able to find small molecule inhibitors. However, RNA-based therapies, together with neutralizing monoclonal antibodies, have been successful, and 2 neutralizing monoclonal antibodies have been approved. But if you look at the cost implications here, we're talking about $8,000 American per patient per year. So it's really expensive, will have to be restricted to high-risk individuals, and of course, not a viable option in relatively poor areas in the world. If you look beyond PCSK9 monoclonal antibodies, numerous promising pharmaceutical leads have failed, and we're talking about over a dozen, costing billions to the pharmaceutical industry. And here are just some examples that I've put together. For example, you hear about HDL as being a good cholesterol. However, agents which elevate HDL have not been effective. They've failed at clinical trials. And what we now think is not about concentration of HDL, but is the function of HDL, the activity of HDL, which is more important. Others being, for example, lipoprotein-associated phospholipase A2. This is an enzyme which produces atherogenic lipids from lipoproteins. Again, a big trial from Glaxo on inhibitors of phospholipase A2 failed at the clinical trials. Cholesterol ester transfer protein is an enzyme which transfers cholesterol ester from HDL to proatherogenic lipoproteins such as LDL. Again, inhibitors of cholesterol ester transfer protein have failed at the clinical trial. Same applies with this enzyme, which is involved in esterification of cholesterol and its storage in cells. Now, this is not surprising, because here we are talking about a disease where agents need to be taken for between 30 to 50 years in the lifespan of an individual. So right from the onset where the disease is seen to the clinical complication. And given the duration, it's difficult to identify sort of the side effects unless one has got genetic evidence, because the enzymes in the genes do other things, in fact, in different parts of the body. And the only sort of target for which we've got genetic evidence is PCSK9. Individuals without PCSK9 don't get cardiovascular disease despite high levels of risk factors. So that's the only case where we've got genetic evidence of loss of function not being detrimental. With others, we don't have that type of evidence in humans. So in light of these issues, people have started looking at other targets as well. So far, I've actually talked about dyslipidemia as one sort of target. The second target which people have sort of looked into, and this also comes with in terms of COVID-19 that Shiv actually mentioned, inflammation. And of course, atherosclerosis, we now know, is a chronic inflammatory disorder of the vasculature. And as Shiv pointed out, inflammation is orchestrated by a range of cytokines, and you've got good cytokines, which are anti-inflammatory. You've got bad cytokines, which are pro-inflammatory. And atherosclerosis, pro-inflammatory cytokines predominant. And I think for about 15 years of my research career, we have worked on lots of different cytokines, because they are promising therapeutic targets. And in terms of therapies, you can think about neutralizing monoclonal antibodies or small molecule inhibitors or soluble deco-receptors. And so we've done a lot of work on cytokines in the past. The problem with cytokines is the complexity and the numbers involved. So this slide shows you the number of chemokines which have been found in atherosclerotic plaques. And of course, they are receptors. A class of cytokines called interleukins. And of course, these other cytokines. And you can see, in fact, that the numbers, in fact, and the complexity that is associated with cytokines. And those in red are the ones for which we have done quite a lot of work in the past. If you take, for example, interleukins, one of the first one identified for which we've got 30 years of research is interleukin-1 beta. The list, in fact, goes beyond interleukin 35. In my laboratory, in fact, we're also working on interleukin 37 at the moment. And this complexity is not just with cytokines. It also, in fact, relates to immune cells. So if you take macrophage, there's not just one type of macrophage. There are about 8 different types of macrophages, and new ones, in fact, are being identified. And the plasticity is quite is tremendous. So a macrophage can change from one state to another one, depending on the environment. So in terms of therapies against inflammation and cytokine signaling, perhaps one of the landmark trial was in 2018, and that was the CANTOS trial. And the CANTOS trial was aimed at individuals, and it's a multinational trial, individuals who already had myocardial infarction, but had a high level of inflammation, and these were on statins. And this high level of inflammation was shown by the presence of high sensitivity C-reactive protein. Now, I'd like to, in fact, highlight here the scale of the trials which are required and have sort of been involved. So in this case, we are talking about 10,000 participants multinationally. And so this one was on a cytokine interleukin-1 beta, one of the first one identified. We've got 30 years of history of research behind it. And it was a monoclonal antibody where 3 doses were used, and 2 of them were found to have significant lowering effects. But again, we're talking about monoclonal antibodies. We're also thinking about sort of manipulating inflammation, which of course can leave individuals more prone to infection. So this type of therapies will have to be restricted to high-risk individuals. But again, shows you that how promise can lead to despair. And this is actually another trial on a very similar principle on targeting inflammation. Similar scale, 10,000 here, about 5,000 patients here. This is called the CIRT trial, the CIRT trial. And the rationale in this trial was that individuals who have got arthritis are predisposed to atherosclerosis. And in fact, in one of the talk, there was a sort of mention about psoriasis and then interleukin 17 axis. And of course, there is a lot of sort of evidence which also shows that individuals with psoriasis are also predisposed to atherosclerosis. Now, in arthritis, low-dose methotrexate was extremely effective. So it was thought, well, it will actually translate to atherosclerosis. So that low-dose methotrexate would also prevent cardiovascular disease in this trial. Unfortunately, that didn't turn out to be the case. The trial was stopped without any success. Now, we actually have published an article last year, shown here at the bottom, where we have tried to actually sort of explain potential reasons why the third trial failed and what's the way moving forward. So, given all this history that we've got behind in terms of pharmaceutical drugs, which of course are extremely effective, one is to take it, but they have got sort of side effects, and of course there have been numerous failures. It's not surprising that a lot of interest has been fueled in the use of natural products, and that's where in fact research in my laboratory in the last 5 years have moved into. With natural products, we have actually got a huge history about its safety profile. However, what we lack in sort of research in natural products is two things. First of all, we don't have deeper mechanistic insight. In some cases, claims are made on in vitro assays. In vivo, in fact, we don't have poor understanding about their bioavailability, but mechanistically, we also understand this in a poor manner. And secondly, lack of large clinical trials. And we're talking here about 5,000 to 10,000 participants, not a dozen participants, and then making a claim. And so this particular diagram, which is taken from one of our articles in Nature Reviews Cardiology, shows the different stages in atherosclerosis and how some of the common nutraceuticals or natural products work. And with the exception of omega-3 fatty acids, our understanding of how different natural products work is extremely poor. And this is an area where we've actually sort of worked quite extensively in the last 4 to 5 years. Now, it's not just one-way traffic. Studies on natural products can give rise to potent pharmaceuticals. And here is just one example. Omega-3 fatty acids, and some of the effects of omega-3 fatty acids are mediated by a G protein coupled receptor 120. And so armed with this knowledge, using synthetic chemistry, a selective agonist of G coupled protein 120 were identified and shown to improve insulin resistance and chronic inflammation. So this actually sort of the collaboration of natural products and pharmaceutical drugs. So what we've done in the last sort of 15 years is developed a range of in vitro and in vivo assays that recapitulate different stages in the pathogenesis of atherosclerosis. And the in vivo studies have been mainly restricted to mice. It's worth pointing out that wild-type mice don't normally develop atherosclerosis, so one has to use genetically modified mice, and the mouse model that we're using in my laboratory is the LDL receptor deficient mice. And of course, this coupled with sort of more cutting-edge technologies allows you to look in a more holistic manner the changes which are occurring at the molecular and cellular levels. So some of the readouts are shown here and extends to hematopoietic stem and progenitor cells in the bone marrow. And obviously, the model allows you to look at a number of things. So this is, in fact, one way of looking at progression of the disease and the effect of an agent on progression of the disease. You can also do regression studies where you develop the disease first and then give your agent to see whether atherosclerotic plaque regress. And you can look at parameters in the blood, lipoprotein profile, cytokine profile, in the arteries and the heart, in the bones, as I mentioned, hematopoietic stem and progenitor cell profile, interaction with other sort of tissues, liver, adipose tissue. And of course, you can employ more sophisticated technology. And one such explosion more recently is single-cell RNA sequencing. So you can sequence, in fact, RNA in a single cell. And this is actually identifying new immune cells that are involved in the development of the disease. At the moment, I'm editing a book on methods in molecular biology on atherosclerosis. It's got 52 chapters and it's going to come out in a latter part of this year. And in this sort of book, we've got sort of not only my laboratory, but world leaders writing about different methods that actually are available. And the one, in fact, I'm just sort of reading today is a single cell RNA sequencing combined with T-cell sequencing, the sort of thing, in fact, which Shiv sort of mentioned. So clearly, in fact, there's an explosion in technology, which is going to further advance our understanding of diseases such as atherosclerosis. So here are some of the agents that are being studied in my laboratory: hydroxytyrosol, a polyphenol found in olives, polyunsaturated fatty acids, flavonols, and again, she mentioned about catechins and epicatechins in green tea. And I've got a program of projects, in fact, looking at catechins in atherosclerosis, phytosterols, and probiotics. And in fact, here are some more recent publication. The one in Food and Function was accepted and published last week. Now, clearly, that's deeper mechanistic insight. Side by side, we did large clinical trials of the scale that, in fact, are used in pharmaceuticals. And here, in fact, we have seen some movement. And this is one trial, the Rijiju trial. The outcome came out again about a year and a half ago. 8,000 patients. These are all on statins, elevated triglyceride levels, and they were given sort of twice daily an ethyl ester of an omega-3 fatty acid, EPA. And again, this was extremely successful, opening up new areas of omega-3 fatty acids, which is a combination of mechanistic insights now backed up by large clinical trials. So that brings me to Ayurveda. And I actually got fascinated when I was asked to give this talk in Ayurveda. In fact, whenever I sort of come to India, at some of the conferences I attend, there is a talk often by Bhushan Patwardhan from Pune, and he gives me some fascinating insight into Ayurveda in terms of applying sophisticated sort of modern genetics technique, in fact, to understand what is actually sort of quite rich, in fact, in our heritage and sort of the Ayurveda, the books, in fact. And of course, Bhagavad Gita is actually one major sort of contributor in this particular field. And so the first thing I did was, armed with the sort of limited knowledge from Bhushan Patwardhan's talk, is I actually sort of did a Google search on Ayurveda and atherosclerosis. And that took me to a number of websites of individuals, in fact, who are trying to sort of sell their sort of agents, or in fact provide what you call armed, scientifically sort of sound, justified sort of treatment avenues. And in fact, so that got me a little bit excited, but I think what put me off is that the majority of this site actually had sort of photos of international models, right? Showing in fact how happy they looked by taking Ayurveda sort of agents which are from India, in fact. And I thought, what's actually gone wrong? You know, we became independent in 1947, in fact, and you know, what's wrong with the Indian models? I mean, you know, the Indians are as beautiful, in fact, they were more beautiful than some of the international models, in fact. And so that got me a little bit skeptical, right, in terms of the claim which was sort of put here. So I then actually went along and did a PubMed search. And of course, PubMed searches are quite sort of good in terms of identifying international sort of quality science, in fact, you know, and I do appreciate, in fact, that funding and so on, in fact, plays a major sort of role, in fact, in terms of the avenue which is chosen in sort of publication. And so you'd have thought, in fact, that obviously well-funded labs will actually sort of try and publish in international journals. And so when I actually took Ayurveda and atherosclerosis as the two terms, in fact, there were only 44 articles. Ayurveda itself, about 6,000 sort of articles. If you look at the individual sort of agents, either in fact in their sort of general name or the scientific name, you actually had in between 300 to about 500 articles. And you compared it with, for example, one of the agents that we are studying, resveratrol, which is found in the grapes, sort of the skin of the grapes, 14,000 articles, catechin, sort of in green tea, and it's obviously quite important part in my laboratory, about 15,000 articles, and hydroxytyrosol found in olives. So that actually just shows you that there is actually more work needs to be done, in fact, which is published, in fact, in international journal. One thing I must actually emphasize here from our studies, from our in vivo models that we have found, is that while natural products, and I think this will extend to Ayurvedic components as well, whilst they're extremely good for prevention, okay, and as a part of a healthy lifestyle, when it comes to treatment, one has to be extremely cautious. What we have found is that in our in vivo models for prevention, the natural products are extremely good. They prevent a lot of different parameters. However, when we go to regression studies where the mice already have the disease, we have found, in fact, that you get subtle changes. Plaque is, for example, stabilized, but it cannot regress plaques. You know, the atherosclerotic plaque is not regressed, which is what you actually find with drugs like statins. So that's, in fact, while there's a lot of mileage with natural products, one has to actually bear in mind that prevention, in fact, is one of the first thing. And then, of course, when the disease has set in, one has to actually take it with pharmaceuticals. It will actually stabilize plaque more. But there, of course, as Shiv pointed out, you need to look at the interaction with pharmaceutical drugs that you're taking, because there could be adverse interaction. So, having looked at that, then I actually looked at those articles which were published, and they are more recent articles, which is encouraging. They are actually dealing with the models that we work on. For example, here, apple protein E deficient mouse, which is another encouraging point. And the third one is that they're looking at mechanisms, which of course is encouraging. So we need more of these studies, which will then, of course, form the foundation of trials. And in fact, if you're going to apply to generally a population in a safe manner, has to be extremely large clinical trials. So summary, atherosclerosis is an inflammatory disorder orchestrated by the action of cytokines. And what I should have told you is current therapies are associated with considerable residual cardiovascular disease together with other issues. There have been disappointing outcome of numerous pharmaceutical leads, natural products, and also, in fact, those that are part of Ayurveda inhibit several atherogenic processes in vitro and in vivo. What we need to do now is more mechanistic studies, getting more deeper mechanistic insight, and eventually large clinical trials. Thank you for your attention, and I'm happy to answer any question that you may have.
Thank you very much, Dr. Ramji. And last time we interacted, we had asked many questions. So I will leave it to other panelists to ask questions. I have a couple of them, but I will do it after everybody else is done with.
Thank you.
Hello.
Yeah.
Hi, sir.
Hi.
So, do you have any plans to do any work on Ayurvedic plant molecules?
We are open, right, for any products. The problem, of course, is in the UK, you're not going to get funding, right? Particularly, in fact, the starting point is actually, as I mentioned, in fact, with the sort of publication, the PubMed and so on, right? It's going to be sort of extremely difficult. But we are actually sort of carrying out studies, in fact, on sort of components which are present, in fact, in the Ayurvedic and herbal sort of agents. So for example, We touched about catechin, in fact, and we've done a lot of work at the moment on catechin. There's two sort of papers that we're writing at the moment. It's one of more promising leads. We've done some work on probiotics. Of course, that's not part of Ayurveda, but punicalagin and pomegranate, of course, and polyphenols there. So we're doing work on that, but not specifically on sort of Ayurveda components. And that's in part because it would be difficult to get sort of funding in the UK from the sort of sources that we get, unless, of course, there's a sort of specific interest or from the Indian sort of companies or Indian sort of organizations which are sort of heavily involved in Ayurvedic medicine. The models are there, the technology is there. It's just, in fact, we need the science behind that. And I think one of the problems that you face with Ayurveda and a lot of sort of this sort of things is that because of resources or because, in fact, there's general feeling of blind faith, right? A sort of a trial of 12 individuals is more than sufficient to convince, in fact, a sort of significant number of people, in fact, to actually make it a profit sort of making exercise. But, you need, in fact, the science behind that, right? In sort of in vitro and in vivo model systems, and then actually sort of large clinical trials to take to the next stage. And that is actually sort of not there, and I think more needs to be done. And so, whilst, in fact, components in sort of Ayurvedic sort of treatments is something which we do work on. We don't really sort of have plans, in fact, to sort of take some of the agents that are sort of shown in the sort of the websites, in fact, into our research sort of going forward.
Sir, is it really difficult to get a license or your approval from from UK to do a work on, especially an extract, as it is not a single molecule, as we are taking an Ayurvedic extract, if it is having a multiple molecules or so. So is it really difficult, or whether is it possible to do it?
No. It's that. So one of the article that I mentioned, which we published it last week, right? There was a combination, right? So what we did was and there was a collaboration with the local company here. That sort of study involved using omega-3 fatty acids in fish oils, phytosterols, and also flavonols combined together. And if you take, in fact, so those actually are broad classes, but there are 15 different chemicals in that study, and we actually sort of managed to sort of carry out that sort of study. So that wasn't a problem. Of course, in terms of publication, right? So the impact factor of that journal is about 4.2. And that's the highest we could go, because one of the questions that often gets asked is that you got this 16 odd chemicals. Which one is doing giving the beneficial effect, and which one, in fact, not? And so it becomes quite difficult to pinpoint, right? But if you're taking an agent, right, and so which is actually a combination, and the agent works, then that's fine. In fact, it depends what the endpoint is. So I just submitted an article last Monday on, I think, one of a very potent agent, which is a probiotic sort of mixture, and that contains 8 different probiotics. So that wasn't an issue. So I think, in fact, mixture is not a problem. I think it's just sort of the grant, so which is required for this, which I think is an issue, in fact, and of course the lack of models. So I don't think in India, for instance, you got many genetically modified mice, which of course is required for studies on atherosclerosis, unless you go to some bigger labs.
So there is always a possibility of joining hands with you for doing a work. Right.
Of course. I'm open for everything. In fact, it's not just related to sort of science. In fact, there's one thing I just always mention is that in this sort of thing is that I actually do a lot of community service, and one of the things we started last April was actually a monthly pratna, singing bhajans, and Lata Ben, in fact, was our first guest singing, and she mesmerized us with her singing. And we've been going, in fact, at international level now with over 200 participants, and we've got one, in fact, coming on Saturday. So there's collaborations with India, in fact, which is which is actually so absolutely wonderful, in fact. And there's so much, as I say, the heritage and our culture, which is behind it, in fact. And it's just, in fact, the Ayurveda lacks that critical science, right, which is required, in fact, to take us to the next level and be, in fact, a leader. So that's the important thing, a leader. And I think that sort of transition needs to be made. It will be made, right? It will take time, but I think something needs to be initiated at this point.
Thank you. Dr. Damle?
I think you're on the mute.
I'm sorry. Yeah, last question. I was about to ask you about funding, which you already answered. What about gut microbiome and prebiotic? Like, I mean, if I'm spelling it right, Akromesia, or group of microbes which actually influence the absorption of fat. So, are you working on any of those factors?
Yeah. So, we actually, I mean, that's an important sort of point to consider when you're taking any of these agents. In fact, is what does the gut microbiota do? And of course, that's where obviously the sort of situation in different population one has to take into account. So, for example, the gut microbiota in the Indian population will be different from that, in fact, in the Western population. Those in the obese will be different from those who are actually diabetic and those who are lean, for instance. So it's a major contributor. And clearly, as I said, the manuscript that we submitted last Monday was, in fact, probiotics, which is actually obviously sort of bacteria, which then actually sort of go into your sort of the gut, in fact, and then modify the gut microbiota in terms of producing beneficial sort of agents, which then actually impact in different parts of the body. And so that interaction is something absolutely important to study. And let me just give you one example, which I think is an important one. So individuals who have got diet which is rich in red meat, for instance, it doesn't apply to me, I'm a vegetarian, but individuals, in fact, who have got diet rich in red meat, they actually, the gut microbiota will produce a chemical called TMAO, okay? And that this TMAO will then diffuse, in fact, from the gut into the artery and sort of cause sort of atherosclerosis progression to be moved forward. So that's an example. There is not the agent that you're taking, but what the gut microbiota is doing, which is then actually impacting. And so in any studies, in fact, that you carry out, you need to actually look at that in more detail, that, what is actually happening to the sort of the metabolism of that particular agent, whether it be prebiotics or any sort of Ayurveda component, or any agent that you're taking, what the gut microbiota are doing, and how you can then actually sort of, that will allow you to produce an actually additional avenue, is your agent plus, in fact, changing the gut microbiota, which could be, in fact, an Ayurvedic sort of formulation, coupled with, in fact, a probiotic or a prebiotic, which actually have more impact than, in fact, each one of them in isolation.
Thank you very much. Moderator, if there are no additional questions, we can ask Vice Chair Dr. Manoj Kalur to conclude, giving conclusion remarks on all the lectures.
Sir, one question to Vamana Rao, sir. Sir, is any issue with the ethical clearance in that present study?
So we have not approached for the ethical clearance. We need to approach, and we have to take an initiative, and some hospital should come forward to do this study, because nobody believes it. Believing it is difficult. Everybody, moment you say it is plant extract, they say no, it doesn't work. It's you believe it, but it doesn't work. That's how they turn it down. That's the biggest question. There should be some belief from a doctor and a clinical practitioner. Then only it is possible. At least it should be used as a first aid and see how it works. And then only you can prove that it is working. But if a step forward is not taken, it is not possible.